Hello.
Okay, so our first presenter is Felix, who did his research with Victor Odo. And good luck, Felix.
Thank you so much. Hello. Once again, good morning to you, friends and senior members. My name is Felix Mensah, and the topic of my presentation is detection of chilling sensitivity in summer squash seedlings of different genotype. And my mentor is Dr. Victor Rudolph, whom I'm privileged to have here in our midst this morning. And my program was sponsored by the Pierce Foundation. Summer squash, which is scientifically known as Cucurbita purple, belongs to the tropical and subtropical areas, and also comes from the Cucurbitaceae family. And some of its members, or members that belongs to this group, are the melons, the pumpkins, the cucumbers, and several others. And it has a temperature growth range or growth requirements range between 24 to 29 degrees centigrade. Low temperatures is a growth limiting factor to most tropical plants and therefore causes chilling injury. Chilling injury is a physiological disorder of plants or plant organs caused by exposure to low temperature but non-freezing temperatures. In harvested summer squash fruit, chilling injury is manifested as pitting, shriveling, necrosis, darkening, and even causes or enhances decay. Chilling sensitivity depends on genotype, the genotype of the plant. As we see here in the pictures, we have two different cultivars. That is the yellow, which is determined by the dominant homozygote B, and then the green color, which is also determined by the recessive B gene. And here what we observed was that when the fruit were exposed to a lower temperature of 7, the yellow cultivar, which is determined by the dominant B gene, express stronger or chilling damage, whilst in the green did not express or express less chilling damage. Whilst when the temperature was raised to 14 degrees centigrade, we observed that there was no chilling damage in both cultivars. So the question we ask ourselves is that, is genetic difference in chilling sensitivity between the yellow and green cultivars manifested at the early stages of the plant development? And the hypothesis was that the first, there is a genotype determined relationship between fruit and seedling response to low temperatures. And the second was that this chilling response can be evaluated by non-destructive spectral method. And therefore, the third is that if the hypothesis of the first and second are true, it may allow selection of chilling tolerant cultivars at the early stages of the plant breeding process. And our main objective was to compare the behavior of the seedling of the yellow and the green genotype under chilling and non-chilling conditions. And our specific objective here was to determine the effect of chilling stress on the root length of the green and yellow cultivars of summer squash seedling after exposing them to low temperatures. Secondly, to determine the chlorophyll fluorescence of the cotyledon of the green and yellow zucchini seedlings after exposing them to low temperatures. And thirdly, to measure the autoluminescence in the cotyledon of the green and yellow seedlings, that is after exposing them to the low temperatures. And finally, to measure the cotyledon color in the seedling of both cultivars after exposing them to low temperatures. So in our exercise, there was 2 trials. In the first trial, what we used was the hybrid to represent the green cultivar, and we used the Goldie to represent the yellow cultivar in our first trial. But in the second trial, we expanded the varietal base to 6, 3 of the green cultivars and 3 for yellow cultivars. And with the green cultivars in the second trial, we used Baraket Laria, and 8 balls. And in the second trial, we repeated Goldie, Sabrine, and then we also used 1 ball. The seeds were germinated in the dark at 27 degrees centigrade for 4 days. After the seeds germinated, we exposed them to 7 or 14 degrees centigrade according to the treatment for 3 days. The seedlings were then transferred to light for 1 day at 20 degrees centigrade. And in the first trial, we measured the illuminance as well as in the second trial. But in the second trial, the illuminance we measured was as doubled as in the first trial. So as you see in the first trial, the illuminance was 7,000 lux, and in the second trial, it was 15,000 lux. And in this case, our factors were the genotype and the temperature, and at least 10 replicates of each variety, that is to the temperature. Then the experiment was repeated twice. Some of the instruments that we use in measuring our parameters are the fluorometer, which is commonly known as the Handy P machine, and then for measuring the chlorophyll fluorescence. And we also use the chromometer to also measure the color in the cotyledon. And also we use the in vivo vision system, which is commonly known as the IVIS machine, in measuring the autoluminescence. So the results for our first trial, what we observed in the root development was that when we exposed the grain, that is the grain cultivar, to 7 and 14 degrees centigrade, we observed that the root development there was no significant difference between the 2 temperatures. But when we also exposed the yellow cultivar to the same temperatures of 7 degrees and 14 degrees centigrade, we realized that at 14 degrees centigrade, the root had some effect, and the effect was even stronger at 7 degrees centigrade. So we went on to measure the chlorophyll fluorescence, which again here I must emphasize that when the bars are low or the fluorescence expression is low here, it means the chilling damage is stronger. So what we observe here with the green cultivar, that is the hybrid, was that at 7 degrees centigrade, the fluorescence or the chilling damage was higher than at 14 degrees centigrade. And even when we come to the yellow, that is the Goldie, yes, at 7 degrees centigrade, we observed that the chilling damage was even greater or stronger than in the green cultivar. So again, we continued with our measurement, and then we measured the color of the cotyledon. And here, there were 3 factors that we used. That is the hue angle, the lightness, and the chroma. And in using the hue angle, the observation was that after measuring the color in the cotyledon, they all had similar color, which was the green color. It was very similar in all the temperature treatment. And then with the lightness, which determines the lightness of the greenness in the cotyledon, the observation was that in the yellow at 7 degrees centigrade, the color was lighter than the rest of them. Even at 14 degrees, it was still lighter than in the green cultivars. So the yellow cultivar or cotyledon was lighter. And with the chroma, which determines the dullness or the brightness of the leaf color. And what we observed was that at 7 degrees centigrade in the yellow cultivar, it expressed more dullness in the cotyledon than in the rest of the treatment. At 7 degrees again in the green, yes, the dullness was there, but it was more stronger in the yellow cultivar. So we went ahead to measure the autoluminescence, which is the photon emission in the cotyledon, and this is usually as a result of the lipid when they are oxidized in the tissues. And here what we observed in the graph is that when the autoluminescence or the photon emission is increased, then it is as a result of chilling damage. So what we observe here in the yellow cultivar was that at 14 degrees centigrade compared to 7 degrees centigrade, 7 degrees centigrade emitted more photons in this aspect. But it was rather opposite in the green cultivar, which we observed that at 14 degrees centigrade, the chilling damage, which is the photo emission, this parameter which was used, was greater than at 7 degrees centigrade. So it became very interesting for us to know what was really happening. So we expanded the varietal base, as I indicated, in the materials and methods to 3 of the green and then 3 of the yellow. But we used the same 7 and 14 degrees centigrade in our treatment. And what we observed in the root development was that at 7 degrees centigrade, in both green and yellow cultivar, we observed a similar trend of the root development, even though 8-ball at 7 degrees centigrade had a higher root development. But on the whole, when you observe, we observe the yellow and the green cultivar, we observe a similar trend. As well as when we also exposed them to 14 degrees centigrade, it was still similar. Therefore, there was no significant difference between the two cultivars at this temperature. So we went on to measure the chlorophyll fluorescence. And in this aspect, again, we observe a similar trend. That is, when we compared the 7 degrees centigrade in both cultivars, that is the green and the yellow cultivar, we observe a similar trend in both cultivars. And again, at 14 degrees centigrade, again, it was, even though at 7 degrees, we observe the chilling damage at 7 degrees compared to the 14 degrees. But when we compared both cultivars at the same temperature, the trend was similar, and therefore there was no significant difference. So we went on to measure the photon emission, that is the autoluminescence. And to our surprise, again, it followed a similar trend. Therefore, at 7 degrees, when we compared both cultivars, that is the yellow and the green cultivars, there was no significant difference in this aspect also. So in our discussion, we said the temperature of 7 degrees apparently caused chilling stress in the seedling of summer squash. And this agrees with Linus. This agrees with Linus, Raab, Soviet, and Morris, who stated that the biggest intolerance to low temperature is found in young plants or young seedlings. And the retardation of seedling root, that is the radical growth, was shown as an indicator of chilling stress. And this agrees with Raab, Soviet, and Kang, who evaluated the chilling injury in cucumber seedlings as the inhibition of linear growth of the radicals after chilling. Again, the decrease in chlorophyll fluorescence was another marker of chilling injury development in the seedling in agreement with Yu et al. On the other hand, the relationship of autoluminescence of the cotyledon with chilling stress was less straightforward, as we observed in the results. And in the first trial, the differences in chilling sensitivity between the yellow and the green genotype was manifested in the behaviors of the young seedlings, as we saw in the root length, the cotyledon color, and in the chlorophyll fluorescence. However, this trend was not consistent in the second trial, as we saw. Almost everything was very similar. And when the seedling of both yellow and green genotype, or the cultivars, it suffered from chilling stress. And we believe that this could be possibly due to the differences in the post-chilling growing condition, as I illustrated or showed you in our earlier stages, where we illuminated the first trial and the second trial, but we increased the second trial by 2, because the first one was 7,000 lakhs and the second was the 15,000 lakhs. So we believe maybe it will be possibly because of this thing, and this is the light intensity. And therefore, we suggest, or we think, a further fine-tuning of the experimental system is needed in this case. And these are my references. And I would also like to express my appreciation to these wonderful people. First of all, to the Pierce Foundation, who is led by Trevor Pierce. This man is really important to me. He has therefore taken, or this foundation has taken responsibility for all my financial obligations so far as this program is concerned, and therefore it's really important to acknowledge them. And also to the international school. I cannot express their professionalism in their work with words, because these people are really fantastic. And also to my mentor, who is my father, and my supervisor as well. This is a very humble and a very patient man. I really thank him so much. And also one very important person I would like to also acknowledge and show appreciation is Dr. Haya Friedman, who is the head of our department. This woman, for her enormous support in carrying out this experiment. I thank her so much. And also to a senior member also in the lab, that is Anton Fenech. He also assisted very, very well in carrying out this exercise. Also to Dr. Yakov Vinokov, who is also a senior member in the lab with all the assistants to this exercise. And finally, to Dr. Harry Paris. This is the breeder who assisted us with the seeds of the summer squash in carrying out this project. Also, I also want to acknowledge Anna Para for her enormous assistance also for this work. And I say thank you, and may the Lord richly bless them all. Thank you.
During your introduction, you showed us some very nice pictures of the effects of chilling injury on developed fruits.
Yes.
But then the experiment is looking at stress tolerance of seedlings. So I'm wondering, can we draw conclusions about the effects of stress tolerance of seedlings on the storage conditions or the shelf life of matured fruit?
Actually, our observation, thank you very much for the question. It is a fantastic question. What we intended to do was the observations we had from the fruit, that is from the literature, which indicated that at lower temperature, or the BB, that is the BB dominant gene, which is responsible for the yellow color in the fruit, therefore had some implication on the chilling damage when exposed to lower temperature. What we wanted to do is that to use this idea to find out from the seedlings if they will show some kind of correlation, that is when they are exposed to these same temperatures at the early stages, so that we can use that to predict that this is how, when this seedling at this stage behave, this is how the fruit will also behave. So thank you so much. Hello, Mom.
What do you think is the B gene doing? So how come the color is correlated to heat or cold stress?
Actually, in the literature, or let me say further readings and the observation we saw was that the BB genes, for instance, in the green cultivar, that is the green, let me say the dominant one, had less tocopherol. And we know tocopherol as a natural antioxidant in the chloroplast, which is responsible for the chlorophyll. And this tocopherol was less in the yellow cultivar, but in the green cultivar, which had the BB recessive gene, had higher tocopherol in the chloroplast. So therefore, we think it may also have some, because when the fruit were measured, that is, the tocopherol level was measured in the fruit, this was the observation made. But in our case, we did not go on to measure this tocopherol level in there. So we think probably maybe it may also have some implication. Thank you.
You started with two cultivars, one yellow and one green, and you showed a picture of the damage caused in the low temperature
Yes.
to the yellow cultivar.
Yes, please.
And then you went and did the second experiment with 6 different cultivars, 3 yellow and 3 green.
Yes.
Did you? There is a possibility that yellow color does not always segregate with chilling damage. Did you check to see if all the 3 yellow cultivars are also more sensitive to chilling damage in the fruit?
Actually, if I got the question right, I say the genes, that is the B dominant gene, which is responsible for the yellow color of the fruit. Therefore, as I earlier on responded to the question to Rivka, I think this tocopherol is, like I said, is a natural antioxidant which protects the chloroplast.
I didn't want your theory. I just wanted to know if you checked or somebody else checked that all the 3 yellow cultivars are more sensitive.
Okay. I think now I get it. Unfortunately, we did not check.
If I can interfere,
Yeah.
I can answer.
Yeah. Okay.
I don't know. I need permission.
But I think we did not.
Permission is granted.
Thank you.
So, in fact, all yellow cultivars, if they are if the fruit is fully yellow, they have this chilling sensitivity problem. All of them have the dominant B gene, and the sensitivity, because all of them don't have normal chloroplasts. Or it is the matter of chloroplast development. That is why they are all yellow, and that is why they are all sensitive.
Another question, you were talking about significant difference or not significant difference. And you were showing graphs with
Yes.
the standard.
As a confidence T.
But did you do any?
Yes, we did.
statistical-
Yes, we did a statistical analysis, and then we used the confidence T to determine this.
So when you didn't mention that it was significant, then it wasn't significant? Because there were some results where you had a very high standard error bar.
Yes, those were, especially for instance, in the root development, when we compared the green to the yellow, it showed significance, especially when we compared the green to the yellow. And this was, we used the confident T to analyze this as well.
Any more questions?
Okay, thank you.
I'd like to introduce you to Ricardo, who did his research with Rivka.
Okay, hello ladies and gentlemen. Welcome to our today's presentation. I'm Ricardo Nagasa. Today I'm here to present a project entitled Identification of Conditions in which silicific acid is active in extending the shelf life of sorghum plants, especially in the tide leaves, under the supervision of my mentors, Dr. Rivka, who has been supporting me from the start of the project to the end. I was sponsored by the first foundation here, which is Felix also introduced, is very important. So in our today's presentation, I'm going to present on with the highlights of the project. Is just the highlight of the project is the first one is to compare the young plants and matured plants sorghum leaf senescences under dark-induced senescence, and also leaf chlorophyll concentrations, as well as the amount of silicon that are extracted in dry leaf, and also silicon activities under stress conditions, under drought and salinity. And also, lastly, I'm going to present senescence-related gene expressions. So when I start with my project overview, So as all we know, the element silicon is a quantitatively major mineral organic constituents of the higher plants, which especially in the angiosperms, it is highest level or accumulation is high, especially in the monocot compared to those of the dicots. One of the monocot is the sorghum plant, which is known to accumulate the high composition of this silicon from 2 to 3% in its dry tissue, so that this plant is considered as a median for silicon accumulators, especially from these monocot groups. In fact, the general trend is just in monocot, the amount of the silicon is just from 1 to 10%, but sorghum is one of the most important crops, which is agricultural important, which is store about 2 to 3% of silicon in its dry weight. So silicon is also used to delay the senescence process. It also helps in improving water use efficiency. Again, it is also important in alleviating of biotic stresses, like as I've highlighted, especially in drought stress and the salinity stresses. Moreover, this sorghum drought weight, chlorophyll level, and photosynthetic rate stay higher, especially when there is an accumulation or treating of this silicon compared to untreated plant species. Again, this sorghum, silicon, it helps to delay drought-induced senescences in sorghum, especially by inhibiting ethylene biosynthesis, which is a hormone upregulated during leaf senescences. Moreover, this silicon supply is correlated to delay dark-induced senescences in detached leaves of sorghum that's I've already reported, and also in Arabidopsis, especially by increasing the cytokinin biosynthesis. That means if there is much more accumulation of this silicon, there is an upregulation of these hormones, so that it helps to reduce the rate of senescences in the leaves of the plants. Here, sorghum mutant tape, that is non-silicon accumulating plants, contain about 40 times less silicon when compared with that of the genotype, which is not accumulating the silicons. Here in this case, we have a sorghum mutant that we already isolated in our laboratory, that we had used it, which is defective to silicon channels. That means this is a gene that is channeling of the silicon from the soil uproot through the roots to the plant, so that there is a defect of these genes, so there is no much more accumulation of this silicon in the mutant tapes. So the lower amount of this silicon accumulation in the mutant leaves enhances earlier senescences. Due to the less accumulation of these silicons, there is an earlier senescences of the mutant leaves compared to that of the wild tape leaves. So even though there is a trend of earlier senescence of the mutant, this phenomena is not a continuous phenomenon. Sometimes it is not a consistent. There is a variation. Due to this, we have a kind of hypothesis. Just the delay senescence is not a consistent phenomena in the two genotypes. Due to this, we hypothesized maybe there is an environmental interaction or physiological condition which may influence the reaction of sorghum to salicylic acid reaction. So that depending on our hypothesis, we have three specific objectives. The first one is to compare the rate at which the wild and the mutant tape sorghum leaves senescent, especially under dark-induced senescence. The second objective is to identify plant growth conditions. That means if there is any influence of growth conditions like stress condition of drought, salinity, or photobreed, or other stress conditions, maybe if there is an expression of these silicons under certain stress condition, and also the last objective is to identify the molecular mechanisms that affect the silicon-induced senescence. So if I start with the first objective, that is to compare the rate at which both the genotype, that is the mutant one, and the wild type leaves will be senesced and the dark-induced senescence. Just the materials and the method is we used sorghum wild type and the mutant plant leaves under were used for these studies. Just data were collected from a young and a mature plant grown under normal environmental condition. Just there is an appropriate normal environmental conditions that were already grown in the greenhouse. And the senescence was induced by placing these sample leaves in a plastic container maintained in humidity condition, especially the 100% humidity condition. And we kept the sample leaves on the average of from 7 to 9 days, and we checked the senescence of the leaves. So the other thing what we did is chlorophyll extraction. Just what we did is we extract from the leaves the chlorophyll. Here tissue samples were frozen at approximately -80°C following dark-induced senescence from the same leaves. And then the frozen tissue cut into small pieces. Then we grounded it and mixed it with 80% aqueous acetone solution. Then after that, finally, chlorophyll concentration were measured by using spectrophotometer. And the last one, what we did is just silicon detection by charring. That is the silicon amount of silicon that exists in the dry leaves. What we did is leaves were oven dried at 60°C, and then we cut it into pieces. And the cut leaves were charred into ash by increasing the temperature to 580 for 24 hours. And then we take the weight again. And then at the same time, we rinsed this charred ash powder with 1 molar of hydrochloric acid in a double distilled water. And after that, finally, we oven dried the sample for 48 hours, and we take the weight of the samples. Then what we found, especially from the first one in comparison of the senescences and the chlorophyll concentration. Sorry. So as I've told you, we compare the matured plants and the young plants. The first one was the matured plant. And if you see the senescence, especially we found in the matured plant senescence, the minus silicon plant leaves, that is the mutant one, senesced earlier when we compare to the plus silicon genotypes of the sorghum plants. However, if you see the chlorophyll concentration from the same leaves, just there is a higher significance concentration of chlorophyll in the case of wild type 6 leaves when we compare to the rest of the leaves, including plus silicon in the second leaves, that is the flag leaves, and minus silicon plant leaves. So that here there is no any difference in chlorophyll composition, whereas this one has a higher chlorophyll concentration in the wild type. Okay, what we did, as I've said, we did also extracting of the amount of silicon in the dry leaves just after we dry the leaves in the oven. Just this amount of silicon accumulated in dry leaves was calculated as a % of acid insoluble fractions per leaf dry weight. That means we calculate we change the ratio of the acid insoluble fraction to that of the dry weight into %, and we express this number in %, so that we assumed that the acid insoluble fraction in the leaves is said to be the amount of silicon that was uptaken by the plants during the plant growth. So if you see from this one, almost in the wild type 6 leaves, that is, there is about even more than 3.5% of silicon was found in these 6 leaves, the younger leaves. In general, there is a high accumulation of silicon in wild type leaves compared to those of the mutant leaves, which have less than 2% in their dry weight. The second one is just we see the comparison, as I've told you, from the young and the mature leaves. In the young leaves, we did also senescences, but we couldn't find any differences in both plus silicon and the minus silicon plant sorghum genotypes. There is a uniform senescences in the leaves. However, if you see the chlorophyll concentration, still there is a higher concentration of chlorophyll in the case of mutant and the wild type 6 leaves compared to the second leaves of mutant and the wild type. So that here, in both of these, there is no any difference in the chlorophyll concentration in the young leaves. Again, there is no any significant difference in the senescence of those leaves in the younger one. Again, we did the amount of silicon dry leaf from those younger leaves. Then we find that there is a higher, again, high accumulation of silicon in the case of wild tepi when we compare to that of the mutant tepi, even though there is a little bit variation among the two. So here there is no any difference in the accumulation of the chlorophyll. I mean, it is even more than 2.5 silicon in the dry leaves. So just what we conclude from this comparison of the matured and the young leaves is here silicon accumulating genotype had higher concentration of chlorophyll, indicating that the senescence is lower than non-silicon accumulating or the mutant plant species. However, as I have told you, there is no any influence of the silicon, especially in the case of younger leaves. So matured sorghum leaf senescence is lower than the younger leaf. However, there is no any influence of silicon, especially in these younger leaves. That is the conclusion drawn from the comparison of the younger and the older leaves. And the second objective is to identify plant growth conditions in which silicic acid is active in extending the shelf life of the tied leaves. That I have said, we expose the sorghum plants to different stress conditions, and we saw if there is a real the same expression or activities of silicon under stressed condition. Just here, the question that we should have to raise is there any senescence-related function of silicon plant growth under stress conditions that, as when we compare to the previous one, under which the plant is grown in normal condition, and the expression is especially, of course, silicon helps to retard senescences of the leaves. But if we expose the plants to stress, what will happen? Is there any function of these silicons? Just from the previous report, what has been reported is, especially if you see accumulation of silicon in the plant leaves, especially in the sorghum, upregulates. This is polyamines. It is a kind of a group of amino acids, so that the upregulation of amino acids blocks ethylene biosynthesis, which is a hormone that enhances the senescence of the leaf, so that there is a blocking of leaf senescences if there is accumulation of the silicons. And on the other hand also, it upregulates cytokinin biosynthesis genes. So if there is an upregulation of these cytokinin genes, this contributes for what? A retardancy of the leaf senescence. That is the report. But for in our case, we tried to see a stress grows, especially the first one is the drought stress. What we did is we carried out drought stress experiment by exposing this both wild type and the mutant leaves to shortage of irrigation for 3 weeks. Then the sample leaves were collected from these stressed plants after 21 days. And the result indicated that it is indicated the same trend is that the minus silicon plant leaves senesced earlier compared to that of the plus silicon plant genotypes. And again, if you see the chlorophyll concentration for the same leaves, just there is a higher chlorophyll concentration in the case of wild type 6 leaves compared to the rest of the leaves. Even though there is yellow in color still, there is no significant difference between this wild type 2 mutant 2 and the mutant 6 leaves. But there is little bit a difference in the magnitude of the chlorophyll concentration. But there is no significant concentration between the two leaves. Here we did also amount of silicon in the dry leaves. We extracted silica from the dry leaves. Just what we found is that still there is a higher accumulation of silicon in the case of wild type leaves compared to those of the mutant leaves, which is more than 1.5%, especially in the wild type 6 leaves. And the other one is we did also drought recovery after we exposed the plants to drought. Then we we did drought recovery by replacing these stressed plants to a normal irrigation condition. That we saw if there is the same trends will happen in the leaves. And just what you will find is there is a highly significant difference, especially in senescences, and the minus silicon is really faster in senescences compared to that of the plant silicon plant genotypes. And if you see the chlorophyll concentration, however, there is no significant differences between these wild type 6 leaves and these minus silicon second leaves of wild type in the mutant. However, here these leaves have very low concentration of chlorophyll compared to the rest of the leaves. Still, we did again the same thing for amount of silicon, the dry leaves. Just we found here there is a high accumulation of silicon, especially in the case of wild type 6 leaves compared to the rest of the mutant leaves. But there is no any significant differences among the wild type second leaves to that of the mutant type leaves in the chlorophyll, amount of silicon in the dry leaves. Then what we did is another stress, that is salinity stress. Just we tried to see if the salinity stress may affect the activities of silicon in leaf, delaying leaf senescences. Just what we did is sorghum seedlings were treated with 100 millimolar sodium chloride and exposed to salinity stress for 3 days. Then we took the sample leaves and kept in dark induced senescence. Then we haven't seen any effects of silicon under these conditions because here there is a uniform senescences and the leaves, so there is no any difference in the senescences of the leaves. However, still they have difference in chlorophyll concentration. Here, the mutant leaves and the wild type 6 leaves act the same way, and that they have the same concentration in chlorophyll compared to the rest of the leaves. Just we did also amount of silicon in the dry leaves, and we found the same trends in which the wild type leaves have high accumulation of chlorophyll, which is more than 3% compared to the rest of the leaves, that is the mutant leaves, which is less than almost 2.5% in the dry weight of the leaves. Then the other stress condition, what we did is we did plants were grown under long and short days, just in the phytotron rooms. The sorghum wild type in the mutant seedling is grown under the phytotron or photoperiod for 6 hours to 18, that is a short day, and 18 to 60 is a long days. And just we see the comparison of this long day and the short day for both genotypes, if there is any effects of this long day and short day photoperiodism on the function of these silicons. And just what we have seen is the long day mutant type and the wild type both leaves senesced slower than that of the short day plants. Short day genotypes. That is both the wild type and the mutant. Still, the chlorophyll concentration have the same trends in which the long day wild type and the long day mutant type leaves have higher chlorophyll concentration compared to that of the mutant leaves in the short day and the wild type in the short day too. And also we did amount of silica in the dry leaves. Now we found, however, that still there is a higher accumulation of silicon in the case of these wild type long day and the short day leaves compared to the mutant leaves of the plants. So that here it is even more than 3% of silicon accumulation in the wild type leaves in both short day and the long day plants. So the general conclusion we drawn from this experiment is that silicon delays senescence in the tight leaves, and also the effect is significant, especially in the older leaves, as I have already presented. The wild type in the mutant young leaves senesced in a similar rate, so that there is no any difference in the senescences of the younger tissue. The chlorophyll quantification doesn't reflect yellowing of the leaves. That means in some cases the leaves look like yellowing, but they have higher concentration of chlorophyll, so that the chlorophyll quantification doesn't reflect in yellowing of the leaf. However, environmental stress like drought and the salinity didn't change the trend of silicon influences on the sorghum plants. That means retarding of the senescence of the plants. Lastly, just the last objective is to identify molecular mechanisms that affect the silicon-induced delayed senescence. Just what we tried to look gene expressions in both genotypes just what we did is expression level of genes along the senescence pathway suggests increasing cytokinin biosynthesis with silicon exposure. That is already what has been reported. Then we tried to find out the IPT gene, that is isopentyl transferase genes, that is especially the gene inhibiting senescence, and the SAG gene, that is silicon associated genes, senescence associated genes expression. However, even though we tried to see the gene expression, unfortunately the result was not successful. So it gives us a direction for further studying, because we couldn't find any expression of the genes and two genotypes. So to summarize, the whole concept of the project is just what we have seen is mutant or minor silicon leaves were senescent earlier than wild type silicon leaves. This might be due to the higher accumulation of silicon in the wild type plants compared to that of mutant type plants. Also, younger tissue were not influenced by silicon treatments, so that as I have presented, there is a uniform senescence, and also in most cases they have the same chlorophyll concentration. Also, there is higher concentration of chlorophyll in wild type compared to mutant leaves. This could be as a result of maybe the antagonistic effects of the silicon on senescence of the plant leaves. So, as I have reported, it retards the senescence of the plants. Again, most of the stressed plant leaves senescent faster than plants grown under normal conditions, as I have already presented in the first one. So, those are the summaries of my projects. Lastly, I would like to convey my thankful for my mentor, Dr. Rivka, who has been supporting for a long period of time. Really, I want to appreciate her, and also Professor Alon, international school head and the coordinator of the research exercise, and Shula Bulum, lab attendant. She's really helping me in the, during my work activities, and also other members, Michael and Niria, who were supporting me in the laboratories. They were a group members in the laboratories, and the Pearce Foundation, who were sponsoring or granting me a scholarship. That is very interesting thing, is that, as Felix said, and also international school. Of course, I have no words to say thankful for the international school, because we are here.
You have no
If
time also.
Yes, I have no time also. Okay, thank you very much for that is the end. Thank you very much. Thank you. Okay.
I have a question. How is silicon getting into the plant? Usually it's not in the form of silicic acid. So how in normal growth condition, or maybe I'm wrong, maybe there are some acids of silica in the soil and it's absorbed into the... And
Okay.
how did you grow the plants before you took samples?
Just the plant were already, it is a mutated plant, and the wild type
No,
plant
how
is
did
that.
you grow them? Did you feed them with silicic
No,
acid?
not,
No?
yes. They are already mutated. The silica, the
No.
plant is already
Not
mutated.
mutated or not mutated.
Okay.
Did you feed them with silicic acid
Not,
so they absorb it from the soil?
yes. Because,
So there is some form of acid in the soil of silicates, and that's how it's absorbed into the plant, I guess.
yes, of course, because silicon is, of course, it is a dominant mineral nutrient, it's the soil component, so that the plants are exposed to the soil so that they can absorb the silicon from the soils. And that is the wild type, and the mutant one is mutated so that it doesn't uptake much of the silicons from the soil.
It does or it does not?
No, it doesn't.
It does not uptake? It does not
Yes,
take?
it does not uptake because the gene is already I presented in the first slide. The gene is
The mutant?
yes,
Okay.
yeah.
May I ask, the difference in silicon content between the mutant and the
Wild type.
normal wild type, is it that level of the difference that you expected? Because it was said that in the introduction that it is 40 times more or whatever, and you observed, let's say, 3 versus 2% or whatever, or I misunderstood something, or the difference was not that great as you promised at the beginning.
Yeah, of course.
Was it so? Was it the difference that you expected to get?
Of course.
To little?
Okay, thank you, Prof. Okay, normally I have presented just in the sorghum plants, they accumulate from 2 to 3% in their dry weights. So the accumulation of this silicon in the sorghum weight is in the range of this. However, in the monocots as a general, it is from 1 to 10% in the dry weight. But here in my presentations, most of them are in the range of 3 to 1, where the mutant leaves have lower concentration, even though there is a variation. Even most of them are less than 2%, whereas the wild type leaves accumulates more than 2% in most cases. But it is not a uniform trend. That is what we hypothesize. If there is the same trends or influence of some silicon reaction to the sorghum plants.
Yes, it was much higher than we expected.
Of course.
I wonder if you check the senescence during growth, not in detached dark induced leaf, because for sorghum, that's the most important thing. I mean,
Of course, normally.
Do you know anything about normal senescence in the mutant versus the wild type?
Normally, we didn't check the senescence in the normal growth conditions. Just we did it after the tied leaves. But under the normal senescence, of course, it is maybe due to the aging of the plants that started to senesce it. In fact,
No, but
we
I
didn't.
wonder if it has an effect.
We didn't check
on,
it. Yeah.
Well, on the normal senescence, not on detached.
Yeah, we didn't
That
check
would
it.
be interesting.
Did someone else check it? Do you know if anybody else in the lab checked this normal senescence?
If I remember, like in the first objective, you did all your measurements on like flag leaf area and the sixth leaf. Coming to the second objective, you were doing on the second leaf and the sixth leaf. So, is there any specific region shifting from flag leaf to second leaf? Because flag leaf is the more important leaf in the cereals, which is a grain filling leaf. So, is there any difference in changing the leaf?
Of course, but the difference is just flag leaves is what we, the name of the flag leaves just after the sorghum plant produce inflorescence. The last leaf next to the flower is just called flag leaves. Of course, there is maybe a difference because the plant is in a different ages. The rest one is just when the plant is mature enough on the young stage of the plant is. But for the flag leaves, we did when the plant is much old enough, when it produces inflorescences. That is the only single experiment we did for the flag leaves.
You said that the white type and the young mutants had the same senescence.
No.
The young mutants and the white type, you said the senescence were the same, like there was no difference. Is that true?
The young mutants and of course, in most of the cases, the younger leaves were not uniformly senescent in plants of the younger. But in the mature case, there is a difference in the senescence again. That means the mutant leaves senesced earlier than that of the wild type leaves in the mature plants. But in the younger plant, we couldn't find any senescence differences.
Okay. You also said you were saying something about slow. The senescence was slower in the white type than in the. Well, how did you determine that? How?
That is, of course, a nice question. Just in most of the cases, the senescence is slower, especially in the wild type leaves compared to the mutant one. That is even our project, our hypothesis. I mean, the higher accumulation of silicon in the leaves of the wild types helps to senescence slower compared to that of the mutant leaves. That is why the mutant leaves senesced earlier, due to the lower accumulation of silicon in the leaves tissue.
That's in the older leaves of young plants. In the younger leaf of young plants, you didn't see it.
Yes, we didn't see the difference in the younger leaves.
Between the age of the leaves, not the age of the plant.
Of course, yeah.
You said that the yellow color was not correlated to the amount of chlorophyll. Can you elaborate what do you think is the reason that what can cause yellowing without reduced chlorophyll?
Normally, what we have seen is just in most of the cases, the leaves look like, under the senescences, most of the leaves look like yellow in the color, but they have still higher chlorophyll concentration compared to the leaves. Those look like green color and dark green color. But I don't know maybe the logic behind why it has higher concentration of chlorophyll when it look like yellow, but higher in chlorophyll concentration than the green one. Maybe Dr. Rivka, if you know something.
That there was a real problem with the quantification of chlorophyll, remember? And then we changed the method
Of course.
for the chlorophyll quantification because the leaves are senescing, they lose weight, and then when you weigh them, it doesn't really reflect the amount of chlorophyll. Still, when you did the area calibration, it was just an estimation, and I think our estimation for the chlorophyll content is wrong.
Okay.
And actually, what we see with the eyes, with the camera, is a better quantification.
So simple apparatus that can measure chlorophyll, you can use them, I guess, for
When we
determination.
got to that point, it was too late in the project.
Okay, thank you very much.
Okay, thank you.
Okay, good morning. I was working in extending the shelf life of banana yellow stage with Haya Friedman as my advisor in the Volcanic Center. It's working. Yeah. Okay. According with FAO, there is a 45% of fruit and vegetable losses in the world, and most of the main reason in developed countries are is in the consumption, in the consumer, and the main reason in the developing countries is about in the processing. An estimation of fruit and vegetables loss in Israel. We have the banana is one of the most highest lost, around 18% in the supermarkets. The commercial practice after in banana is first the banana is harvested in yellow in green stage, and when it has 3/4 of the heat size, and then it's treated with ethylene, and then it's developed in the supermarkets. And here we have some problems, even if the banana is over ripening, and it becomes a problem in the packing houses, or even if the banana is still green and it becomes in brown, and it has the customer has a problem. About the mechanism of ripening banana, as we know, banana and tomato are climateric fruits. They have a peak of respiration before they get ripened. And the changes in the fruit ripening are in the cell wall modification, in the carbohydrate metabolites, pigment changes, phenolic compounds, volatile compounds, and organic acid changes. The lab has identified some genes related with the banana ripen, and they belong to the matbox genes. And these genes are similar to the ring genes in tomato. They are also related with the ripening in tomato. A knowledge-based approach in dealing banana and ripening in the lab. How we know, the lab has silenced some genes belongs to the mat box, and by RNA interference, and they got good results in delay the ripening of banana. And also they tried some modification in the atmosphere to delay the ripening banana. My main goal was finding treatments that will inhibit the yellow stage of banana. And the specific goals were to check the effect of cold treatment on ripening, to evaluate the effect of 1-MCP, that is an ethylene receptor antagonist, on ripening, and to examine the effect of controlled atmosphere. To develop also tools to evaluate the stress period to its visual detection. In my experiment, I utilized three kinds of bags to study the effect of the modified atmosphere. I study the normal bag that is made with polyethylene. I check the effect of the step bag that is made with polyethylenamide, and I check also the rope bag that is made with cast polypropylene. And also I check the effect of the cold. I did two treatments. I put cold before ethylene and cold after ethylene. And I study the effect of the inhibition of the ethylene with the 1-MCP. That is how we know the 1-MCP has a similar molecular like ethylene, and it inhibits the perception of the ethylene in the banana ripening. So here are my treatments. The first one is the control, that the one is like the commercial practice is working. And the two next treatments, here are the order how I treat the treatments. I put cold and then ethylene. In the third treatment, I put ethylene and then cold. And to evaluate the inhibition of ethylene, I put ethylene and then MCP. And then I did 4 treatments to evaluate the modified atmosphere with different packaging. So I put ethylene and then I put the banana in normal bags. I put the banana in a step bag and then in the camera with ethylene. I put ethylene and then the banana in a rope bag. And in the end, I put the banana in the rope bag and in the chamber with ethylene. To evaluate the effect of all these factors, I measured the total solids with refractometer. I check the pigment changes with Minolta, that give me the color of the banana, with delta E, that give me the chlorophyll content, and with IBIS, that is give me the chlorophyll fluorescence and the lipids luminescence. And to check the firmness of the banana, I measure with penetrometer. And for the respiration and the ethylene production rate, I did with gas spectrometer. The first graph give us the results of the treatments, that how we can see all the treatments are better than the control, than the commercial practice, all reduce the production of ethylene. And in the production of CO2, we can see that cold treatment has a lower production of CO2. This treatment, it was first, I put 24 hours in ethylene and then 24 hours in cold at 6 degrees. And also we can see that the packaging has some effect in reduction of the CO2 if we compare with the control, with the commercial protocol. When I measure the firmness of the banana, I found that the treatments with cold and the packaging were the best treatments. The banana is still firm. And when I measured the chlorophyll content, in the beginning, after 4 days of the treatment, I found that the cold and ethylene was highest, the chlorophyll content. It kept greener. But after 8 days, the better treatments were ethylene and cold, and ethylene and MCP. They keep the highest greener, the more greener, and also Stepak and rope. They were the best treatments to keep the color green. And it's related with Minolta. Minolta gives us the color of the banana, and we have almost the same results that when we measure the chlorophyll content, that the best treatment was ethylene plus cold and Stepak plus ethylene. They were the treatments which keep more green the banana until the end of my experiment. And here we have some interesting result that still the modified atmosphere is working well. It produced sugar. The sugar content was higher with the modified atmosphere, and we see here that until here we saw that the cold was, both of the treatments were good, but here we have a difference, a clear difference between the treatments with cold. The treatment when we put ethylene and then we put cold, the cold block the sugar content, the production of sugar content. And when we put cold plus ethylene, the content of sugar it was higher. So for us, the potential treatments are still the modified atmosphere with the packaging, and in between the two treatments with cold, the potential treatment is the cold, and then when we put ethylene, because the banana it will be tasty. So for this reason, I developed a second experiment to see how long we can put the cold treatment. And I did a second experiment. I put the banana in 1 day cold, 3 days cold, and 6 days cold. And also I evaluate in 3 different temperatures, 6 degrees, 10 degrees, and 12 degrees. And luminescence, how Felix explained us, give us the photon emission by the oxidized lipids. So how we can see here, at 6 degrees, we can see the more photon emission than the 10 degrees and 12 degrees. That it means the plant tissue is more stressed at 6 degrees, and it has more photon emission. And if we compare 1 day cold, 3 days cold, and 6 days cold, we can see that the best treatment when we can see really the photon emission is at 24 hours in day cold, because 3 days cold or 6 days cold, the tissue is already dead, and this is why we cannot see the photon emission of the oxidized lipids. And then with the Abyss machine also, I measure the fluorescence of the chlorophyll, and I use the wavelight of 430 and 420, that is the excitation of the chlorophyll. And I got the same results, almost the same result that the photon emission, that the autoluminescence, that the best, when we can see the stress of the plant tissue is at 6 degrees. I cannot get a higher fluorescence of the chlorophyll at 10 degrees and 12 degrees. And also, in 3 days and 6 days, I cannot see any, it's not high, the fluorescence of the chlorophyll. So this is a way to develop a new tool to see how long we can put the plant tissue in cold. And we can see here there is a higher correlation between 420 GFP fluorescence or also with luminescence. And as a summary and conclusions, all the treatments reducing ethylene production on the 8th day of the shelf life. Cold after ethylene reduce respiration rate and keep the firmness and green color. Until here, it will be a potential treatment, but decrease in sugar content, so the banana is not going to be tested. The third conclusion is that MCP maintain green color. It mainly affecting the color, enable sugar accumulation, but did not maintain the firmness. So MCP didn't maintain the firmness. Control atmosphere, step back and rope, keep high firmness, green color, and enable the sugar accumulation. So this is, it could be the potential treatments with control atmosphere. And also, but we think the potential treatment is the cold before ethylene. When we put cold and then we put ethylene, the banana keeps firmness and enable sugar content. That is, the banana is going to be tasty, and this is the potential treatment. And also we can realize that the luminescence and chlorophyll fluorescence are transiently induced due to the cold treatment. It can serve as a tool to evaluate the duration of the cold treatment. So to summarize everything, when I inhibit the production of ethylene with one MCP as a receptor antagonist, we realize that it changed mainly. There is a changes in pigments. It blocks the pigments change, but not in cell wall modification and in carbohydrate metabolism. When we work with modified atmosphere, we block the respiration, and we block the pigment changes, cell wall modification, and carbohydrate metabolism. But when we try it with cold, after ethylene, it blocks the 3 factors. But when we put ethylene after cold, this is what we think is the potential treatment. The pigment changes, it's blocked, the cell modification is blocked, the banana keep green and firm, and it becomes sweet. So this is what the main result of my experiment. This treatment would be the potential. And what I am suggest is to analyze the effect of the cold at molecular level. And also, I couldn't do the phenolic, what happened with the cold or the modified atmosphere with the phenolic compounds, volatile compounds, and organic acid changes. And this is all what I do. I thank you for all this year. For me, it was a great experience, and I am so grateful for the teachers that I have here in the university. Thank you very much. Thanks. Questions?
In materials and method, you said that from the day 0 to day 19 there was different kinds of treatment. Was it for the same experiment you need, same sample? Or
yes, it was the same sample, and I measured all the parameters at day 0, after 4 days, after 8 days, and after, in the end, of, the banana was still available to measure two treatments were still until 19 days for the same experiment. Yes.
Same sample also, banana, same banana.
Same banana.
It's not exactly the same.
Oh, sorry, not exactly, but they comment from the same day. Okay, it was not exactly the same banana because but the same batch.
Same batch.
Yes.
What is the control environment? So what do the bags do?
In the bags, we call it the modified atmosphere because there is differentiation of the exchange of the gases between the banana and the atmosphere. So they have some different materials, the propylene, polyamide, gas polypropylene, and also they have differentiation in the microperforation, macroperforations. They changes in the change of the gases.
Do you know what's the atmosphere when you?
No, we have to do that. We thought later that it was better to measure the respiration rate inside the bag. I measure, but outside the bag. When I take the banana, I put in the bottles, but I didn't know in the bags, in the same bag. And it will be better, the results.
I think that the most important, the most interesting from my viewpoint is the effect of the inhibition of, let's say, the effect of cold. Do you expect, so at 8 days it was still with very low sugar. Do you expect it will ripen or later, or it is just arrested at this stage?
It's already the banana was yellow, and it was yellow, so it's not choice to wait until develop some sugar, more sugars.
So it is a kind of chilling injury, you can
Yes.
say. Can we?
Yes.
Yes. And could you elaborate what was or what could be the mechanism of this injury? Because you managed somehow to overcome it. Or maybe I'm not clear.
I don't understand it.
No, but you said that, so it is. We said that it is a chilling injury and it will not ripen normally. But then when you apply 1 MCP, you do manage to override to correct.
That's not. These are all different experiments. MCP is alone. Cold is alone.
Sorry. So then I don't understand because am I right that there was cold and 1 MCP?
No. It's not this treatment. No, it's cold after ethylene and cold before ethylene. This is of the two treatments about the cold. And to study the behavior of MCP, it's with ethylene. It's the ethylene and MCP.
Yes.
Ethylene and MCP.
So I was confused, but ethylene and cold, you have ethylene.
I put ethylene and then I put cold. And cold, it's blocking all the mechanism. The cold after ethylene is somehow block the effect of the ethylene. The cold, it block the effect of ethylene.
Ethylene after cold.
And ethylene after cold, it's able to develop.
Correct.
That's correct only in sugar content. This is what we think is a potential treatment.
Yeah, so that's what I had in mind. So sorry for, let's say, for dragging it into some direction, but would it mean, I will say by myself, yes, that this mechanism of cold effect was inhibiting the ethylene production? Because if you added ethylene, you managed to correct it.
Yes, It's possible. What we see, it's at least the sugar content is correct with ethylene.
Yeah.
But in the other way,
in spite of giving the cold treatment. So probably.
yes, it will be. Yeah, this is what I suggest to study in the molecular level what is the effect of the cold in the multiple genes.
Can you show your model again?
No, it's not this. Which one? This one?
The last one. So, according to your model that you found, you were mentioning in the beginning that in the lab there's RNAi lines that have reduction of some mass oxygen, right?
Yes.
So, how do you think they would be affected by all these treatments, cold and MCP and the controlled environment? Do you think they'll react the same like the wild type plants, or will they react differently?
I think they will react different because they silence some genes related to it. So they already got results with this kind of silence genes, and they got good results. They delay the ripening of banana.
Okay, but I'm asking about yours. I know that you showed that.
Yes.
But I'm asking about these different treatments. How would these antisense plants react to these different treatments? Would they be
What I think
the
is
same as wild type, a little bit more delayed, or would they react?
what I think is the cold is is somehow a transcription factor. So this is going to be delay the ripening of the banana. This is what I think.
I am Nirmal from Nepal. Today I am going to present my research exercise work entitled Differential Pathogenicity and Host Specificity of Soft Rot Pectobacteria. This work was conducted under the guidance of Dr. Iris Ideda from Department of Ornamental Horticulture in ARO, Volcani. Okay, let me first introduce you with the soft rot Pectobacteria. These bacteria belongs to the genus Pectobacterium. They are gram negative, rod shaped, facultative and necrotrophic bacteria which belongs to Enterobacteriaceae family. They were previously in the Erwinia genus, later they were moved to Pectobacterium. Among the different species of Pectobacterium, these are the most studied Pectobacterium species: Pectobacterium keratoborum, Atrosepticum, Beta vascularum, Wasabi and Pectobacterium aeroidearum. These all Pectobacterium has the capacity to secrete exoenzymes into the intracellular space of the host, and they degrade the host cell wall. Because of this, they cause soft rot disease in most of the economically important crops such as potato, carrot, cabbage and many ornamental plants, and now they are becoming threats to these crops. This Pectobacterium has broad host range and they are distributed all over the world. They can attack about 35% of the angiospermic orders and more than 50% of the monocot plant orders. And they are isolated from plants, soil, water and even from the insect host. And among the different species of Pectobacterium, Pectobacterium wasabi and Pectobacterium atrosepticum are most host specific. Pectobacterium beta vascularum has narrow host range, whereas Pectobacterium keratoborum, subspecies keratoborum and brasiliense has broad host range. And till today, we do not have any effective control measures for this Pectobacterium, and because of this, this bacteria is ranked among the top 10 bacterium most effective infectious bacteria. This is from the paper published by Isse and his team. Here they isolated 47 different isolates of Pectobacterium keratoborum, subspecies keratoborum from different geographical location and from the different host, and based on 16 years rRNA sequence, they found that isolates from monocot host clustered in one clade in the phylogenetic dendrogram, whereas the isolates from dicot host clustered in the another clade. It means monocots were clustered in one place and dicots were clustered in one place, and the similar kind of results were obtained from AFLP as well as from the ITS PCR banding pattern. So, based on this work and based on this similar kind of work, we were interested to see what will be the host specificity of different Pectobacterium strains, but based on their fitness to their host. So, we hypothesized that Pectobacterium keratovorum strains isolated from monocot are more compatible with the monocot host, whereas than dicot host and vice versa. For this, we set our objectives to study the interaction and host specificity of Pectobacterium strains isolated from monocot and dicot plant host, and we set 3 specific goals. First one is to study the disease development in cabbage and calla lily in the presence of monocot and dicot specific strains, to study the antimicrobial activity of plant phenolic extracts, and then to study differences in virulence gene expression of tested strains in the presence of plant phenolic extracts. So, for this we first did virulence assay in 2 different hosts. One is monocot and monocot was calla lily and cabbage representing the dicot, and we selected 4 different isolates of Pectobacterium PC1 and PC16 as monocot isolate and WPP14 and 1692 as dicot isolates. And we did virulence assay. From the virulence assay, we see that when we infect the host leaves only with distilled water, then we do not see any infection in the leaf, because it was the control with water, so we do not see any disease symptoms. Whereas when we infect the calla lily leaves with the monocot strains PC1 and PC16, then we see nice soft rot disease symptoms you can see in the picture. Whereas when we infect the dicot host cabbage with the same monocot strains, then we do not see typical soft rot symptoms, although we see some infection, but the infection did not develop to up to the soft rot symptoms. Whereas in case of dicot strains WPP14 and PCB 1692, we get opposite results, where these dicot strains were unable to infect the monocot host, and whereas in case of cabbage, these dicot strains were able to cause typical soft rot symptoms in the cabbage which is dicot host. So, from these results we see that monocot isolates, monocot host, and there is development of typical disease symptoms. Likewise, dicot strain, dicot host, there is development of typical soft rot symptom, whereas the reciprocal of these 2 combination did not develop typical soft rot symptoms. So, based on this finding, we were further interested to see what will be the antimicrobial activity of these 2 hosts in these strains. So, for this now afterwards we use only 2 Pectobacterium strains, PC1 representing monocot isolate and WPP14 representing dicot isolate. And for this, first of all we extracted plant phenolics from these 2 hosts. We first collected leaf samples, we cleaned them and disinfected them with the sodium hypochlorite, and then we grinded them in the presence of liquid nitrogen, and then we added 1 ml extraction solution, which is the combination of double distilled water, methanol and acetic acid in the ratio of 5:11:1. And then once we get the phenolic extract, we use hexane for cleaning and removing all the unwanted chloroplast as well as other compounds. And we perform growth response of these phenolic compounds in 2 selected strains PC1, PC16 and WPP14, and we get these results. This is the growth curve of PC16, where we treated this bacteria with different concentration of phenolic extract from calla lily as well as cabbage, and we see with the higher concentration of phenolic extract from both calla phenolic extract and cabbage phenolic extract, we see great reduction in growth of this strain. However, once we decrease the concentration of the phenolic extract, then we see the growth was not affected much. And when you see the same effect in another strain, dicot strain WPP14, we see the similar kind of results. It means with the higher concentration there was reduction in the growth, but once we decrease the concentration of the phenolic extract, then the growth was less affected. And in overall, we can see cabbage phenolic extract has better antimicrobial activity than the calophenolic extract. So, to sum up results from this assay, we see some antimicrobial activity of this phenolic extract. However, we did not see any host specific antimicrobial activity which we see in the virulence assay, where monocot was favoring monocot host and causing disease agent, but we do not see such host specific antimicrobial activity from the growth response curve. So, further to explore host specificity, we were interested to see what will be the effect of this phenolic extract in the other virulence determinant of Pectobacterium, and the virulence determinant of Pectobacterium are exoenzyme activity as well as biofilm formation. So, first we go with the exoenzyme activity, and in exoenzyme activity we mainly focus on three exoenzymes: pectidase, polygalacturonase and proteinase, and we get these results. In PC16 strain, which is I remind you, this is the monocot strain, we see that the exoenzyme activity of PC16 was significantly reduced by cabbage phenolic extract, that is from the dicot host, whereas the exoenzyme activity was not significantly reduced by the calophenolic extract except with the activity of PRT. But in case of WPP14, we get the opposite results, where exoenzyme activity was significantly reduced by calophenolic extract, whereas with the cabbage phenolic extract the exoenzyme activity remains same as of control, except the activity of polygalacturonase, where the activity was even enhanced by the cabbage phenolic extract. So, from here we see differential response of two tested strains, that is one monocot strain and one dicot strain towards the extract of their respective host. So, here we see monocot isolate, phenolic extract from dicot host, significant reduction in exoenzyme activity, and reduction in exoenzyme activity means no suitable situation for disease development. Likewise, dicot host, phenolic extract from monocot host, then there was significant reduction in exoenzyme and no favorable condition for disease development. Once we see this differential response, which corresponds with our finding from the infection assay as well, we were further interested to see the biofilm formation in the presence of this phenolic extract, and we assayed biofilm formation and we got these results. In PC16 bacteria, the biofilm formation was significantly increased by calophenolic extract, whereas with the cabbage phenolic extract there was no significant difference in biofilm formation. Whereas in case of dicot strain WPP14, the biofilm formation was significantly enhanced by cabbage phenolic extract, but not by calophenolic extract. And let me remind you, biofilm formation is necessary for successful infection to the host. So, it means here we can see cabbage phenolic extract is significantly significantly enhance the biofilm formation in PC16. It means this is the perfect condition for disease development. So, to sum up the results from this biofilm formation assay, we see monocot isolate and phenolic extract from monocot host, then enhanced biofilm formation and perfect condition for disease development, and same with the dicot strain, and this finding also corresponds with our virulence assays findings. So, once we get these results, then we were interested to see what will be the differences in the virulence gene expression in the presence of this phenolic extract So for this, citing from the literature, we selected 6 important virulence genes, gene PEL and PES, which were related to exoenzyme secretion, gene HRPL, which is related to secretion system and elicits hypersensitive response. We selected gene EXPI, which is the regulator of quorum sensing in Pectobacterium, and 2 transporter genes PSTA and ARF, and we use REC-A gene as housekeeping gene. And for virulence gene expression assay, we first grow bacterial strains in LB medium in the presence of plant phenolic extracts. The plant phenolic extract used was non-lethal, and then we extracted RNA from those bacterial cells after 8 hours, and then we prepare cDNA, and then we quantify the mRNA by using qRT-PCR, and we got these results. In case of PC16, the gene PEL was significantly upregulated by Cala phenolic extract, whereas in case of dicot strain WPP14, the expression was upregulated by cabbage phenolic extract. It means we see differential response in gene expression, which we previously see in the infection assay as well as biofilm formation and as well as in exoenzyme activity. Whereas in case of another gene PES, which is also the exoenzyme gene, we do not see such differential response, whereas in this case the expression was significantly downregulated by both Cala and cabbage phenolic extract. In case of another important virulence gene EXPI, which is related to quorum sensing signal generator, we see similar kind of trend as of PEL gene, where in monocot strain it was significantly upregulated by Cala phenolic extract, whereas in case of EXPI gene in WPP14 it was significantly upregulated by cabbage phenolic extract. So we see the differential response as in gene PEL. Whereas in case of other 3 tested genes HRPL, PSTA and ARF, in PC16 their expression was significantly upregulated by both phenolic compounds, and in case of WPP14 their expression was significantly upregulated by only cabbage phenolic extract with the exception of gene PSTA. However, in these 3 genes we do not see any differential response which we see in other genes PEL and EXPI. So, to sum up these results, we see reciprocal expression of exoenzyme related gene EXPI, we see PEL, we see reciprocal expression of quorum sensing system gene EXPI, whereas we see a similar expression pattern in other related genes. So, based on the results of all the assays which we conducted, we came to the conclusion that phenolic extract from cabbage and calla lily were found to have some antimicrobial activities. Pectobacterium strains were host specific, such that monocot isolates prefer monocot host and they were more virulent towards monocot host. Likewise, dicot isolates prefer dicot host and they were more virulent towards dicot host. However, we cannot say this unless and until we do some further works such as fractionation of phenolic compounds present in both plants and see what is the difference between phenolic compounds in between in them, and as well as to see the expression or to see differences in virulence gene expression of more quorum sensing related genes, quorum sensing regulated genes and other virulence genes. I would like to thank International Scholarship Fund for providing scholarship without whom I will not be here, and I would like to thank Pierce Foundation for organizing different events during the study period, and I would especially like to give thanks to my mentor Dr. Iris Ideda for all the support and suggestion. As well, I would like to thank Mr. Janakraj Joshi for his continuous guidance and support during the research period. I would like to thank all my lab mates and all other batch mates. Thank you.
So I saw that you have you chose several genes that are related to quorum sensing.
Yeah.
And some of them were induced by the
extract,
and some of them were not.
Yeah.
Is it possible, based on the knowledge that exists in the literature, to build a model why why there is a specificity of the response, or it's not, you don't have enough data yet to do that?
If I understand the question, when we go through the literature, then we see that with the phenolic extract from non-host plants, and in most cases we see the down regulation of the virulence related genes, because they have some antimicrobial activity. Whereas when
No, let me clarify the question. Let me clarify the question. You have a set of genes.
Yeah.
They know, you know from the literature where they work and how
Yeah.
they act.
Yeah.
You didn't show a model how they act, but I'm sure that it's known in the literature.
Mhm.
So is it possible to show, to explain from what you know in the literature why, based on what you found, why this cabbage extract do not work on calla, or do work on, is it, or it's not enough, you don't have enough the whole picture of the quorum sensing? I mean, can you imagine in your mind how all the genes are related to quorum sensing?
Yeah, because EXPI gene is related to quorum sensing system, and when EXPI gene is expressed or upregulated, then this quorum sensing system gene also helps in enhancing exoenzymatic activity. So it means EXPI or quorum sensing system is regulating the activity of exoenzymes, and these exoenzymes are the one which cause disease symptom. So here we see with the calla, the EXPI gene was upregulated significantly as compared to cabbage. So maybe because of this reason there was high expression of exoenzyme related genes, and there was high expression of exoenzymes, and maybe because of that the disease was much severe with the monocot strain in the monocot host, and same with the dicot strain with the dicot host.
So what is the most significant, what is the gene that is most important for quorum sensing that determine the quorum sensing behavior?
Gene EXPI and gene EXPR are the genes which are responsible for quorum system, and these quorum system regulated quorum system genes regulate other different virulence.
So they are upstream of the whole process.
Yeah, downstream of the other virulence genes are downstream of the quorum sensing system genes.
You were showing the beginning results, showing how each, when you use one strain that was for dicots, how it influenced the monocots, right? And you had the virulence, the amount of virulence, and you were showing these spots.
Mm-hmm.
And the spots were much smaller when you did the cross testing. Does this actually, these smaller spots, they actually mean less virulence, or could you have other interpretations?
Yeah, this is less virulence. They cause disease symptoms, but the disease symptoms was limited. They show the infection, but the infection did not grow as it was grown in case of monocot strain with the monocot host. Monocot strain, and they develop typical softer symptoms, whereas we tried with the dicot strain in the monocot host, and the symptoms was there, but it did not develop as in case of another combination.
We saw that.
Yeah.
You showed that in the pictures, but is it a question of time? If you waited more time, would both spots grow and grow, or is it when you did the cross, would it always stay small?
No, it takes it does not grow as high as compared to another one.
So what is the meaning of this rate of growth? Does it actually mean less virulence, or does it mean something else?
Yeah, it means less virulence. Dicots were found less virulence towards monocot.
That's a sentence you say. I'm just asking what's behind it. What is actually the growth of the spot? What does it mean? The rate of growth of this circumference, what does it actually mean?
It means the you mean the reasons?
Yes, what is actually making a spot grow bigger and bigger and bigger?
It's because of the exoenzymes which they secrete. They degrade the host cells, and because of this the infection grows more. and these are the necrotrophic bacteria. So they wants to first kill the cells, and then they use these killed or died cells. So it's because of the exoenzymes the spore grows increase and increase.
The phenolic extracts you were using, you were taking them from plants that were not infected, right?
No, they were not infected.
Do you think it would be different if you took it from phenolic extracts from infected plants? Would they might possibly have some?
Maybe there will be difference because of the infection. They could further more they could enhance the phenolic extract if they were exposed to the bacteria, or if they were infected with the bacteria, then maybe the phenolic extracts will increase in the plants, and the extract from those plants will behave differently. But we have to do the research for that.
One last question: if you mixed the two strains, the bacteria strains, and added them to each one of the plants, do you think they would behave like the more virulent or the less virulent?
Actually,
the spot grow just like. Do you think it might be?
Actually, I have no idea about that because I
You
didn't.
can hypothesize.
Yeah, we can go further.
In the first graph that you show in cabbage, your treatment of 2020 was higher than the control. Do you know why?
Wait. A lot of yeah.
In the cabbage, the treatment of 20 milliliters was
Here?
higher than the control, and that doesn't happen in the other graph.
I don't know exact reason for this, but I go through the literature and found that there are some microbes which are able to degrade even the phenolic extracts, and they use those phenolic extract for the carbon or the energy source. So maybe because of this reason, and since 20 microliter is not in high concentration, so because of that reason they may have used it as their own benefit and enhance more. I think so.
Okay.
Did you do the columns which handle your bacterial gene? And did you take the column sensing molecule detection of column sensing means the bacteria induce the
You mean AHL?
AHL signals?
No, I didn't do that.
Okay, any more questions? So thank you.
Thank you.
So Iris was very lucky to have two students.
Very good students.
Two excellent students, and
Thank you.
now we're going to hear what Beatrice did.
Okay, good morning. I'm Beatrice Was Gideon, and welcome to my presentation on the topic of beneficial microorganisms to support the health and growth of ornamental plants. I took my research exercise in Volcani Center under Dr. Harris Yedidia, and then my sponsor is Pears Foundation. Okay, ornamental geophytes. These are also known flower bulbs, herbaceous perennials with underground storage organs, like bulbs, tubers, rhizomes. Genus Xantheschia, color lily, is a monocotyledonous tuber plant from the Hariaceae family. This family comprises of about 8 known species. 6 of them are colored estivized species and 2 white species. It's very important in ornamental plant. It's abundant worldwide, and it's cultivated for various reasons. One of them, tuber production, cut flowers, and potted plants. Ornithochelum is a genus of monocotyledonous perennial plants,
and it is found in the family Asparagaceae. Formerly it was in Aethalaceae. It comprises of about 200 known species, including Ornithogalum dubium. The flowers are known for its extreme long-lasting vase life. It means you can store them for a long time. And it has a very bright orange flowers. Therefore, both species are known to be sensitive to bacterial pathogens, including the soft rot, which is caused by Bacterium carotivora. I stress that because our research is based on these phyto and other fungal pathogens. The geophytic lifestyle of these ornamental plants makes them perfect hosts for these soft rot diseases. The reason is very simple. The bulbs and the tubers, they act as the package of starch, and once this starch is degraded, it releases molecules of sugars, which are very good growth substrates for pectobacteria. And then, up to date, there are no effective control measures of this disease, according to Lazzato and Yedidia. So in order to minimize the damage which is caused by these bacterial and fungal pathogens, we initiated an approach of using some of the environmentally friendly products to support the growth of these two ornamentals, Xandeschia and Ornithogalum. And we went for the already commercialized products, including the Green Up. This is plant-based product, and it is from food and drink industry. We have Rhizoctor is from Andama Makteshim Limited, and probiotic mix is from Better Hair. So we hypothesized that beneficial microorganisms or the natural products which were specifically developed to support the plants may improve the plant health in soft rot sensitive crops relative to the non-treated. These growth-promoting products may improve the yield and reduce the development of soft rot diseases in the greenhouse. Therefore, we set the objectives, and the general objective is to improve the growth and development of the two geophytic plants using environmentally friendly products. Our specific objectives, one is to characterize the product effects on the growth and development. The second one is to analyze the development of the bacterial populations or microbiome in the growth substrates. And then the last one is to study the product effects on the plant defense response. Therefore, we set our experiment in Lahishi Experimental Station near Kiryat Gat. We set it in a randomized complete block design with 4 treatments: Bacillus subtilis, and Rhizoctol, Green Up, that is plant extracts, and control, we used tap water. We replicated our treatment 6 times on a plot of 5 by 0.5 meters squared, and we gave them the treatment's designation letters. Letter A to represent control, B probiotic, C Rhizoctor, and D Green Up. This is the experimental design from the start of the experiment up to the end of the experiment. We started with the selection of the tubers and bulbs, eliminating the rotten and the non-healthy ones. So we only used the healthy ones. We inoculated them in our treatments for 10 minutes, and then we planted at a spacing of 10 centimeters on a pallet media. Pallet is known to be a sterile media, and it has a good water absorbent, and that is how it looked like. For the start of the experiment, this is at the end of the experiment. Then We collected our data periodically based on monthly basis, and we used several methods. We took measurements like plant height. We also used photographs. We calculated measurements of the flowers, and also at the end of the season, we took the bulb weight. We went ahead to collect the microbiome data. We collected 3 samples for the 4 treatments. In each treatment, 3 samples, and we replicated 3 times. So we collected around the rhizosphere zone, and we made sure that we were very careful not to injure the roots. Then we sent the samples to the high labs for the analysis. It's good to know what they did over there. What they did is the samples were put in 50 ml tube, and then once taken to the lab, they did DNA isolation. They did amplification using PCR, and then the amplicon products were purified and sequenced using the next generation sequencing. And then the operational taxonomic units as the outputs were then analyzed. So my results, we start with the in vivo, that is from the greenhouse plant growth analysis. And on the x-axis, these are the treatment. We start with the A is the control, B is Bacillus, Rhizoctor, and green up. On the y-axis is the average plant diameter in centimeter. For the first month on January, we found that green up and Bacillus, they were significantly different from the control. For the second month, we found that green up and Rhizoctor and Bacillus, they were also significant from the control. And the last month of March, we found that green up, Rhizoctor, Bacillus shows significant different from the control. What can we conclude from this? That our treatments significantly promoted the growth of the plant diameter in relative to the control. We also checked at the average leaf number. Unfortunately, the results were not significant. We went ahead to look at the mean average root length. On the x-axis, these are our treatments. A is the control, always. A is the control. B, Bacillus, Rhizoctor, and green up. On the y-axis is the average root length in centimeter. What we found is green up significantly increased the root length compared to the control. And also it reflected on the pictures. Whereby we see the length of the roots in the green up, they were much longer compared with the control, and also they were more compared with the control. This is in calla lily. Indubium also showed the same results. We see that in green up, the roots are much longer when we compare them with the control. We also looked at the mean average number of flowers, and we found that the control, the green up showed significant difference when we compare it with the control. So meaning that green up here is significantly contributing to the plant shoot biomass and also the root system growth. At the end of the season, we took the bulb weight and we did analysis. We took the measurement of the bulb weight and the number of bulbs and the weight per bulb. And unfortunately, these treatments didn't show any significant results. We also looked at the plant growth parameters in calla lily, and we found that in our treatments, the control Bacillus, Rhizoctor, and green up, we found that green up and Rhizoctor significantly showed an increased number of open leaves in response to the control. And also, when you look at the average mean plant height, we found that the control, Bacillus rhizoctor and greenup, average plant height, rhizoctor, during the first month of January, it significantly increased the plant height in response to the control. But in the second month, there was no difference. It means that rhizoctor promoted the plant growth of height at the early stages of development. And then we went ahead to look at the microbiome results in the for the two plants. And looking at the taxonomic compositions of the rhizosphere soil microbial communities, we found something common in the two plants, that the bacterial and archaeal groups, they dominated. And the bacteria which dominated include Protobacteria, Chloroflex, Plantomites, Actinobacteria, and two archaeal groups, Thermomachiota, Euchromachiota. These two groups are very important for the plants because they participate in ammonification, the change of organic nitrogen to ammonia. And Protobacteria is very useful in nitrification. It converts the ammonia into nitrates, which are taken up by the plants and used for the growth. And then they went ahead to do a principal coordinate analysis in order to check whether the results are making any sense. And they found that rhizosphere bacterial communities, which are associated with calla lily, they clustered together, as shown here. And they found that the rhizosphere bacterial communities associated with the dubium, they also clustered together. But the funny thing, the results, the treatments, when you look at the B, A, and C, and they didn't show any significant difference. But how come that these ones, they have shown something like clustering together in calla lily and clustering together in dubia? They went ahead to look whether these results are making sense, and that's why they did an OSIM. This is analysis of similarity, and they found it is making sense because the p-value was found to be 0.001, which is highly significant. So what can we get from these results? The results shows us that there is a strong effect of the host plant on the soil bacterial populations, and that our treatment, our beneficial microorganisms, they didn't have any effect. Or they were, what I mean is the effect of the host plant was much stronger than the beneficial microorganism treatments. And then we come to the in vitro screening of the beneficial microorganisms or natural products against Pectobacteria. These experiments, we wanted to test whether our treatments have any effect, like defense effect against the Pectobacteria in vitro. And then we started with the first experiment, which we tested on the defense elicitation. And on the x-axis, these are our treatments. The angriotics, based on Bacillus, PCB3 is the control, positive control, rhizoctol, greenup, and distilled water. What we did here is we dipped the cabbage leaves in each treatment for 24 hours, followed by challenge inoculation of the PCB3. This is a Pectobacteria strain. So what we found here, we found that rhizoctol, it is here, it is significantly provided or shown defense effects against the Pectobacteria in relative to the control. And if you look at the leaf disc, you see the infection area. that here the infection area is less. When we look at the control, is much more. And also, something we found interesting is in agriotics, that instead of providing any defense, it accelerated the infection. And to check, maybe because agriotics is bacteria and it did something on the leaf disc, but to prove that it didn't do something on its own, then the leaf disc that we infected with the distilled water didn't show any infection. And then we went ahead to look at the antagonistic effects. And then here, these are the treatments. We have the root shield, we have PCB3, probiotic, rhizoctone, green up, and liquid broth. So what we did in this experiment of antagonistic effect, we inoculated the treatments, for example, root shield and PCB3 together on the leaf disc, so that we can examine the antagonistic effects. And what we found is that the rhizoctone, again, it is showing significant antagonistic effects against pectobacteria in relative compared to the control. So here it shows also a corresponding results from the defense experiment. And also we found that this time around, green up completely inhibited the pectobacteria. There was nothing from the leaf disc. When we look at the root shield, we found something related, which was related with the agriotics here, that it accelerated the infection area. So from these results, we found that rhizoctone, and as shown common mechanisms, antagonistic and defense elicitations in both experiments. We went ahead to look if these to check these antagonistic effects against pectobacteria, this time around using solid crystal violet pectide. And what we found, these are our treatments: pectobacteria, root shield, rhizoctone, green up, probiotics. And this is the pectobacteria colony forming units per milliliter. So what we found here is green up and probiotics, they significantly inhibited or suppressed pectobacterium growth. There was nothing seen. And it provided, in addition, it provided a long-lasting inhibition effect. Up to 7 days, there was nothing seen in these two treatments. And also we found that the root shield, it significantly inhibited the pectobacterium growth up to, for short time, up to 7 days now it started, the pectobacteria started growing. And then we found that the pectobacteria is the control. 3 days, it was full of pectobacteria. So also this experiment, it correlates also with the defense and the antagonistic experiment which we have done previously. And then we went further to look, maybe these bacterial microorganisms are secreting some of the compounds which are acting against the pectobacteria. So what we did is we filtered the two treatments, probiotic and rhizoctone. We filtered them so that we can remove the microorganisms to use the filtrate itself. So the filtrate acted as the inoculum. We inoculated them, on the leaf disc, and what we found is something very funny, because when you look at these, the all leaf discs are infected. This is the control. You don't see any difference apart from the negative control, which is liquid broth. So what can we learn, what can we say about this? We say that the treatments, the beneficial bacterial treatments, they didn't have any effect on the inhibition of Pectobacterium. So suggesting that maybe even this media, what it did, it induced or enhanced the bacterial virulence. And to sum up is that we found that green up extract significantly improved plant growth, and most of the important plant parameters like plant diameter, flower numbers, and root length. And also we found Rhizoctor significantly increased the opening of the leaves and the plant height as compared with the control. In vitro, we found that Rhizoctor, green up, and probiotic treatments, they acted against the Pectobacterium using antagonistic and defense elicitation effects. And what we learned from the microbiome results is that they revealed the plant modulation or the influence of the Rhizosphere microbiome was in host-dependent way, and that our beneficial soil treatments didn't have any effect on the soil microbiome. To conclude is that this present study, it has strengthened our hypothesis, which we had already said that beneficial microorganisms or biological-based products may increase plant growth and health, both in vitro and in vivo, as it was demonstrated by the two treatments, green up and Rhizoctor, have been shown higher potential of defense against Pectobacterium, and also the green up have been enhanced significantly most of the important plant parameters like shoot biomass and root systems. And also Rhizoctor, we saw it acting on the flower numbers, increasing, and the plant height. So what we found is that no previous report which was encountered on inhibitory or stimulatory mechanisms of these control engines, specifically these two, green up and Rhizoctor. And this could be the first report on growth and defense, defense effects against Pectobacterium carotivora. So this our study now, it forms, it acts as basis for future studies. And now studies should be done on the mode of action of these products in relation to the soft rot bacteria, and also other soil inhabitants like nematodes. And also more work should be done on the calibration, on treatment concentration, intervals of application, and duration, in order to understand the full potential of these beneficial bioinoculants on the plant growth and health. I acknowledge the following people, mostly to my mentor, Dr. Iris Yedidia, for her supportive. And also I appreciate my lab members, including Mr. Janak, he is here. He helped me a lot. Nahum, Mikal, and Nimal, also student colleague. And also to the Shlomit, Medina, and Dr. Shiri from ARO Center for doing bioinformatics and statistics of the microbiome. And also to my sponsors, Pears Foundation, for giving me scholarship, for the International School of Agricultural Science, including Alan Samea. Thank you.
In vivo,
Yes?
why did you collect samples from the reservoir to be measured in the microcom?
Okay. Microbiome. Okay, that's a very good question. Let me go back. So your question is why did we collect samples from the Rhizosphere zone, right? Okay, the reason as to why we collected samples from this Rhizosphere zone is that, first of all, Rhizosphere zone is influenced by the roots. And It's rich. This zone is rich in nutrients from the plant exudates, like amino acids and sugars, which are released from the plant roots. And therefore, these sugars, they act as source of energy or a good source of nutrients for the soil microbial communities. So that's why we get a very wide range of diverse microbial communities around this zone.
Could you please go back to the beginning when you had the greenhouse set up for the experiment?
Yes.
Okay, here you wrote that the experiment or in a randomized complete block design.
Yes.
Could you explain what this design is?
What is design?
This randomized complete block design, what does it mean?
Okay, what this means is that the way we set our experiment, we set it in blocks first, and then it's a method of setting up a experiment, and it means we carried it not in the in vitro, it was in the field, not in the it was in the controlled environment. That's or.
So you said you set it up in blocks?
Yeah, in blocks we have blocks of treatments. For example, when we did the replications, we did it randomly. So each treatment fall on a particular block. Yeah.
Go to the next slide. This is the way it was laid out in the greenhouse?
Yeah, that is how it looked like.
And this is a complete block design?
Randomized. Completely randomized block design.
So where is the block?
The block here, what we are referring to the block, these are the plots. These, like this. The problem I have not shown the whole plot here. But here, you see from somewhere here up to, we have a white tag here. It means that is one block.
What does it include?
What?
That block, what does it include?
It includes a replicate of a treatment.
One treatment?
Yeah.
Okay. Okay, thank you.
You think the microbiome, you showed that it was different and it depended on the plant and less on the
treatment.
Do you think if you take the same plant from different sources and you do this experiment again, you begin with what kind of tissue in order to do these experiments?
What kind of tissues?
Yes. Well, what do you plant here? You plant bulbs, right?
Yes.
So if you take bulbs now from a different country, the
Okay.
same species exactly,
Okay.
and you do the same experiment, do you think you'll get the same set of rhizosphere? Do you think it depends on only on the species, but also where you got the bulbs?
Okay, it's a nice question.
Thank you.
Yes, well, it doesn't depend on the what have you said last one?
Does it depend only on the species, or also where the plants, where the bulbs were grown?
Yeah, it depends with the several factors, not only on the plant species or the plant genotype. It also depends on the location, the site where you collected your soil samples. Yeah.
Okay. Any more questions?
Thank you. Good morning. I'm going to present my seminar on the topic impacts of earthwork byproduct and organic amendments on the wheat development and the biomass of the wheat. My mentor is Jose, and then I found, sorry, I found funded by the this one Johnny Memorial Foundation. Introduction. I am going to introduce my this one seminar topic, the construction process during the urban development, construction of bridges, dams, the excavation of the soil. By the digging of the soil, it generates the huge amount of earthwork byproduct that it has the limited use, and then it needs the urgent solution to reuse that soil for some purpose. Then this earthwork byproduct is it contains low organic matter. Organic matter, it is very important for the plant development. Means organic matter, it helps to absorb the moisture, it helps to increase the water holding capacity, water retaining capacity, and then it prevents erosion, and then it provides the soluble nutrients, plant available nutrients, and then so the earthwork byproduct, which is taken from the lower strata of the soil, it contains the low organic matter, hence we consider this poorly fertile. Then this restoration of the fertility, we have the several techniques to restore the fertility. From the different studies, we found that restoration of the fertility can be done by the adding the manure, adding the organic fertilizer, and then it increases the organic content of the soil, and then it helps to provides the nutrients, plant available nutrients, and it helps to increase the water holding capacity, prevention of erosion, and then several benefit from that. And then this restored soil, we can use this, sorry, this can be used as a substrate for the agriculture, and then we hypothesize in that way, like this earthwork byproduct, it is less fertile, it may affects on the crop development and negatively. By restoring the fertility, by adding the amendments, organic amendments, it gives the better result. We hypothesized or experiment in that way, and then we set the objectives, assessing the effect of earthwork byproduct application on the development and other growth parameters of the wheat, and then assessing whether organic amendment it might improve the crop growing on the earthwork byproduct, means not only the wheat, and then how the potential of the our treatment, how it going to affect on the growth of the plant. And then next going to the materials and methods which we followed. First I like to describe this on site and then design of our experiment. This our study site, it is located in the Le Havre. This is the arid region of the southern Israel, and the soil type is loess, and the average rainfall of this area is 18.8 degrees, average temperature is 18.8 degrees Celsius, and the average rainfall is 306 millimeter. This is the plot design, that plot design, and then it showing the elevation in that plot. And then we have the 4 different treatments. the one is control, and then and another one is the addition of the subsoil, means the earthwork byproduct, and the addition of manure, and then the last treatment is the both addition of both manure and then subsoil. Each treatment consists of the 4 replicates, and then each plat, it is divided into 3 subplats, up, middle and down, to study the effect of the topography, how the topography is going to affect on the crop development. And then the sowing of the wheat, it is done in the November, and the sampling, we collected the samples at the 3 different stages of the developing stages of the plants, and the first sampling is done in the January, and then in that samples we assess the in the field analysis, we counted the number of seedlings, wheat seedlings, and then wheat seedlings, and number of grass seedlings, and then the forbs seedlings, and then we assess the dry biomass of the seedlings. And then the second sample, it is collected in the February, in that we measured the plant height and number of ramets, and then dry biomass of the shoot. We did the soil analysis in that frequent time, and that only one time we did the soil analysis in the February, and then in the April we harvested the wheat. We harvested both wheat and weed, and we harvested the wheat hay, not for the grain, because of the lack of rain in the rainy season. And then for the purpose of the analysis of the whole biomass of the area, to assess the treatment on the biomass, we harvested 1 meter square area. And then for the to assess the plant development, we collected the plant samples, and then we assessed the number of tillers, ramets, and then the dry biomass of that shoot. And we also analyzed the root biomass in the 0 to 20 centimeter depth. I am going for the result and discussion section in this part. I am in the first sampling is done in the January, and then we analyze it for the shoot biomass. It is showing the manure, it is increasing the shoot biomass. We have the four different treatments, like this one is the no subsoil and no manure. Here the we have the manure, but no subsoil. Here we have the subsoil but no manure. Here both manure and then subsoil. We got the result. We got the better result in the manure, addition of manure, and compared to the this one subsoil. Subsoil, it is slightly decreases the shoot biomass. Here with subsoil and manure, it improves the shoot biomass when compared to the single alone application of the only the subsoil. And then the effect of the treatment on number of wheat and then wheat seedlings, we found that the wheat seedlings were high in the wheat seedlings were high in the subsoil, and then the it is compared to the this one control, less in the treatments, different treatments, number of seedlings, but it is maybe that space may be occupied by the weeds. Here the weeds are more in the treatment with the manure, and then this one is the grass seedlings, grass weeds, and then this is the dicot weeds or forbs. It is more in the manure, and then we found that number of forb seedlings quite high here it is showing, but in the later stage we got less, very few this on the dry biomass of that dicot seed. I will explain that later. Maybe because of the development process is slow in the dicots, it has not competed well with the this one grass and then wheat seedlings. Effect of the treatment on the shoot biomass and the plant height, which one collected in the second stage, means in the February, it is showing the result. Here also it is showing the better result in the treatment with the manure treatment. When the organic amendment, it is improving the result, and then we also did the effect test. In that effect test, we got the effect of the topography, and then the manure, and then this one subsoil effect. Those effects were significant. And then number of ramets, both plant height and then number of ramets were assessed. We got the better result in the both number of ramets and then the plant height. We got the better result in the addition of manure, and then there is no significant difference between the addition of subsoil and then the without addition of subsoil. We also did the soil analysis. In that soil analysis, we did the phosphorus and then the nitrogen analysis in the two depths of the soil. One is at the 0 to 20 centimeter, and then the 20 to 40 centimeter. We got the weird result, and then phosphorus is better. It looks quite better in the this one manure treatment when compared to the subsoil and then the control. And then in the total nitrogen at the two depths, we found that the nitrogen content is also, total nitrogen content is also high in the addition of manure, and then application of manure is helping the increasing the available nitrogen. Here we tested the amount of ammonia and then the nitrate. We found that concentration of ammonia is high compared to the concentration of nitrate because of the maybe because of nitrate is it is very unstable, and then it changes from one form to another form, and then ammonia content is high, maybe because of the less water content, because of the lack of rain, moisture is less in that season. For that, when the moisture is high, the nitrogen cycle it is quite rapid, but here maybe nitrogen content it is showing the ammonia is higher when compared to the nitrate. And then mineralization and nitrification. Mineralization means the conversion of the organic nitrogen to the this one nitrate, and then nitrification means the conversion of ammonia to nitrate. Here we found the here it is showing the reduction in the nitrate mineralization process. It is reducing. We did the incubation for the 30 days, and then we measured the mineralization process per day, and we found that it is quite difficult to explain the process because of the this mineralization process. It is quite rapid, and then it changes time to time very frequently because of the several factor, and then soil moisture, temperature, and then microbial activities are all involving in this process. And then mineralization process, it is in the upper strata, it is means in the 0 to 20 depth, it is showing the quite higher this nitrification process in the topsoil when compared to the nitrification in the lower depth, and then it is showing the in the upper depth, it is showing the better result in the addition of manure. Effect of treatment on the soil water content, we analyzed the soil water content in one time, and then this effect is also it is high in the addition of manure. We did the dry biomass assess, we found the better result. We found that the better result in the application of the manure, it is increasing the dry biomass, overall dry biomass of the 1 meter square area, and then the even in the weed biomass is also quite higher in the application of the manure. And then I forgot to mention about the soil water content. We did the effect test of the different factors like topography. The topography, it is effect was very significant, and it showing the water content at the up, it is always less, and then the water content at the down, it is always high because of the elevation. Water goes down, and then the moisture content is low in the at the down, and then it is high. Water content is high at the down, and then it is low at the upper layer, upper subplat, and it is only considered in the middle subplat. Root biomass, when we applied this on subsoil, it may happens like the subsoil, when we add the subsoil, root may go deeper, and then it accumulates more biomass. We may get at the lower depth, but we found at the upper depth, we found the less root biomass in the application of the subsoil. Application of subsoil, it is quite decreasing the root biomass. Analysis of the this one wheat biomass, it is showing the plant analysis. It is showing that the shoot biomass is higher with the application of manure, and then the number of tillers, and then number of ramets, and the number of tillers is, and then the tiller biomass, it is also significantly higher with the application of manure. And then I like to summarize this one, different, because of the different effects, it is showing the different result. It is quite weird result we got, and then but we got a better result. We always get the better result in the application of manure. The subsoil is negatively affecting on this one biomass and then the different parameters, but organic amendments, it is improving the yield and then the different growth parameters of the crop, and then the we get the better result in that. And then root biomass, I already mentioned that because of the addition of the subsoil, that subsoil it may as the less fertility, and then the plant roots, it goes deeper and to find the get the nutrients. So we got the less this one biomass of the roots in that soil. And then I would like to conclude that long duration of study is required for this process. It is the processing of the soil formation and then the mineralization process. It is a gradual process. It is not a simultaneous process. And then everything is considered under the economical aspect. This earthwork byproduct is the cheap material. We can use this as a substrate for the refill the soil, means due to the erosion, and then it creates the percolation in the soil. In our field, we can add this as this one earthwork as a substrate, and then we can restore the fertility by adding the different organic amendments or the different technique of the soil restoration process. We can improve the result. We can use this soil as the amendment for this purpose. I would like to thank my mentor and then my lab mates and all my friends and the Johnny Memorial Foundation for the funding. And then I would like to thank for division for all their support. Thank you all.
Sorry, I'm not from the field. Can you explain just a bit about the subsoil, how it is done in the field?
Okay. That subsoil, what we used here, it is collected from the B horizon of the nearby road construction process. We collected the subsoil from there, and the addition of subsoil was in the, sorry, I forgot to mention that addition of subsoil and then manure we did in the last year, means before this experiment. We did that, and then that subsoil means the soil layer, it contains the different horizon, horizon A, B, it has the several horizon. We collected the sample from the B horizon, means that horizon it contains the less organic matter, but it may contain some leach nutrients, and then it contains the minerals, but those minerals are not soluble minerals, and then they may not be available for the plants. So it takes time when we amended with the organic material. It contains the soil microorganisms. That soil microorganism, by the process of mineralization, like nitrogen fixation and then phosphorus solubilization, all those processes, it may increase the result when the addition of subsoil. We don't know whether it increase the this one result, the plant development, or decrease, but we just check that whether it increase or decrease, how it is going to affect that subsoil. Means it is the lower layer of the soil. From the top soil, it is very fertile, it contains organic matter, so it is less in organic matter. We tested that.
So I saw from your presentation that in some cases, for instance, if you look at the weeds, when you added the subsoil, you had less weeds.
Yes.
In both treatments, even if you added manure, the subsoil contributed to less weeds, if I remember well.
Yeah.
I think that in the other treatment there were more weeds. I'm not sure.
But we don't know the reason that there is a chance to that manure may contain some weed seeds. That subsoil, I don't know how it is going to affect on this one germination.
Anyway, there were some positive effects of the subsoil,
Yes.
from what I've seen, and some negative effects. For instance, the yield eventually did not increase with the subsoil. So what is your recommendation for now, from what you know so far? Do you think it is good to use the subsoil, or you can say right now what you think?
We cannot say this is the, as I mentioned, it is a gradual process of the this one formation of soil.
Because the subsoil, when you added it, there were less weeds.
Yes.
So this is a good effect of the subsoil if you have less weeds in the field.
We tested for the several effects, means the topography and maybe lack of rain. It may not that soil is not homogeneous. Means we did the treatment, but in the field condition it is different. Homogeneous. When we consider the overall result, it is we always get the better result in the application of manure, and then because of several studies also shows the organic amendment, it improves the result. And then effect of subsoil, as I mentioned, it may contains some leached nutrients and then deposited mineral contents. The liberation of minerals after some times it get liberates due to the some microbial activity. It may provide nutrients, and then different plants, that grass, it may needs the different nutrients, and the dicots, it may needs the different nutrients. It is maybe helping the germination. We cannot say whether it is soil, that subsoil is helping to reduce the number of weeds. We cannot say that it needs maybe more studies.
You've been talking about the chemical properties of different strata in the soil.
Yeah.
What about physical properties? Are there any differences in how they might affect your results?
Physical property means we only tested for the soil moisture, and then we did the soil moisture test only once. That moisture, it varies time to time, and then it receives the less rain also. So that is the physical. We are not tested for any other physical properties.
Just think what physical properties might differ.
Maybe the porosity of the soil, and then the different physical parameters of more. This subsoil may contain the more gravels, and it depends on that. We have not tested for the different physical properties of the soil, and we are mostly concerned about the plants, means the plant development. It is a combined work. Jose knows that someone they are doing the some group they are doing the soil properties and the soil aspect. We are mostly looking for the plant development, effect of our treatment on the plant.
No, I know what you did. I'm just wanting to think about other aspects that might influence your results.
It may influence.
Okay, thank you very much.